So we're going to continue on with chapter 8. This is going to be part 2. I split part 1 into 2 segments. We'll see how long this one is. Right now it's going to be 1 segment. So we're going to talk about how we regulate protein synthesis. We talked about some enzymes being controlled at a genetic level. Well, these mechanisms ensure that genes are only going to be active when we need to make the products. Remember, Mother Nature is thrifty. It does not want to waste any energy. So we only want them to be active when we actually need the products. This prevents waste of energy and resources. We've got things like our antisense RNAs, microRNAs, and riboswitches, which actually help regulate this in bacteria, archaea, and eukaryotes. So we're going to look at what's called an operon. These are going to be found in bacteria and archaea. It's going to be a set of genes that are going to work together to regulate something as a single unit. These can be either inducible or repressible. In catabolic processes, we have operons. Those are going to be induced by the substrate, which remember the substrate is whatever we're working on, of an enzyme. And so it's going to code the genes for that, or it's going to produce the enzyme when the substrate is present. And that's the only time it will be produced, is when you need that particular enzyme. We also have repressible operons, and that's going to be an anabolic enzyme. These are going to be turned off by the product, which is going to be made by the enzyme. This is best understood when we are looking for a system to control something through genetic induction or genetic controls. Okay, we're going to look at the lac operon, which is something that bacteria like E. coli have. So we have 3 features of a lac operon. Okay, we have the regulator, and that's going to be the gene that codes for the protein that is capable of repressing or turning off the operon. That's going to be a repressor. We have the control locus. Remember, a locus is basically just an area on a gene. So the control area is what you can look at that. The promoter, which is going to say, hey, this is where you need to start. It's going to signal the RNA polymerase where to start at. And then you have the operator, which is basically like the on and off switch. So the locus, or the area, is going to be made up of these 3 genes that code for different enzymes that are needed to catabolize, which remember that just means break down lactose sugar. This is only going to function or work whenever glucose is absent. If glucose is not there and we need to use lactose sugar, then the cell's energy needs are not being met by the glucose if it is there. Those 2 reasons are when we would do this. Okay, so glucose, remember, is a preferred source of carbon. So if glucose is present, it's going to be easier to metabolize the glucose. If not, we need to use lactose, and so we need to be able to break that down. But we don't want to make lactase, which is the enzyme to break down lactose, if we don't need it. That would be a waste of time. Phase variation. This is a type of variation of the phenotype. Remember, that's the physical characteristics that is heritable. It deals with turning on or off genes, usually describes the ability of bacteria to change part of their surface anatomy that the host immune system actually recognizes. Remember, it's going to recognize it as non-self, so they're able to change that. These molecules change the ability of a bacteria to stick to surfaces as well. So they can adapt to and stick to different environments that they normally would not be able to. So 2 bacteria that are capable of doing this are going to be Neisseria, and that's their production of fimbriae, and then Streptococcus in being able to produce a capsule. Bacteria can also change their DNA by undergoing recombination. Bacteria don't have an equivalent to sexual reproduction. So an event in which a bacteria actually donates DNA from one bacterium to another is where we're going to see some recombination. The end result is going to be a new strain from both the donor and the original recipients. And it's going to depend on the fact that, remember, that bacteria have those plasmid circular DNA. And so they're very adaptable at being able to interchange change those genes. And this is good for the bacteria, but really bad for us because this is how they provide genes for resistance. This is how they pass them along. Resistance to drugs like antibiotics, okay? Or being able to produce a toxin, a metabolic poison. They're able to gain a new nutritional or metabolic capability, the ability to survive perhaps in a high pH. They can also have increased virulence or other ways to adapt to the environment. So it's how they can pick up a new handy trick, in other words. So recombinant, that's going to be any organism that contains the genes that actually originated from another organism. So it's going to contain the original DNA molecules plus the other organism molecules kind of scrambled up or recombined. So horizontal gene transfer, this is the transfer of DNA that allows organisms to acquire new genes, which are not coming from the parent. That would be vertical gene transfer. So this is what we were talking about a moment ago, and there are some other ways to do this. This usually involves plasmid DNA or chromosome fragments, little pieces of chromosomes. Remember, plasmids is that circular DNA. They have their own origin of replication, so they can be copied or replicated independently of the bacteria's normal chromosome. These usually only have a few dozen genes. They can carry, again, those useful traits like antibiotic resistance. Even chromosome fragments from a bacterial cell that have undergone lysis or rupturing can be involved in gene transfer. So even if you destroy that particular plasmid, sometimes those genes can still be copied. Okay, and so then those fragments have got to be integrated into the bacterial chromosome in order to be copied and passed on. So here we've got our 3 modes of gene transfer in bacteria. This is based on the mode of transmission. This is going to be our conjugation, transformation, or transduction are the 3 ways this can occur. Conjugation is going to require the attachment of 2 related species that are going to form a sort of a bridge with that we call a pilus, which is going to actually transport the DNA. Transformation is the transfer of basically naked DNA with no special mechanism or mode of transfer. And then transduction is the transfer of DNA from one bacterium to another by the bacteriophage. And remember, a bacteriophage is that virus that actually infects the bacteria. Look a little more closely at conjugation, okay? This is that mode of genetic exchange where a plasmid or other genetic material is going to be transferred from a donor cell to a recipient cell via that direct connection, which is going to be a pilus. Both gram-positive and gram-negative cells can do this. Bacteria can do this through conjugation. It's just a little different for each. Gram-negative conjugation, the cells are going to have the donor is going to have the plasmid. We're going to call that the F or fertility factor, which means it has the genes to make that conjugative pilus, that extension to transfer the genes. This is called the F positive cell, fertility positive cell. The one receiving the genes will have a recognition site on the surface, and this one we're going to call the F negative or fertility negative cell. So the pilus is going to grow out from the F positive cell attached to the F negative cell surface and draw the 2 cells together. Now, in gram-positive conjugation, basically there's just an opening created between 2 cells that are nearby, that are adjacent. The replicated DNA is going to pass across from one cell to another. Conjugation is a conservative process where the donor bacteria retains or conserves a copy of the genetic material being transferred. All right. All right. And so this can confer things like antibiotic resistance, resistance to antimicrobials like heavy metals, being able to make those virulence factors like make a certain toxin, a certain enzyme, or an adhesion molecule to help it stick to a particular surface, things that are helpful for the bacterium for survival. So here's an illustration here of the F factor in transfer. This is the F factor for E. coli. The donor cell here, this is your F positive, is going to make a copy of its F factor. All right, it's going to transmit this through a pilus. It's going to make a bridge and it's going to transmit this to that F negative cell. All right, and so it's going to turn an F positive cell capable of producing a pilus and conjugating with other cells. We have what we call high frequency recombination donors. That means they are able to do this a lot. The plasmid DNA is going to actually then become integrated with your F positive donor chromosome. It's going to be copied and then be transferred to the recipient cell. Some chromosomal genes will get transferred. Some of the plasmid genes may not be transferred, but you can see here in this picture, we're going to copy the important parts from one to the other. So now let's take a look at bacterial transformation. So we're going to have a chromosome that is going to be released by a cell that is lysed. Remember, lysed and loosened. It's going to rupture. It's going to break into small fragments, and they're going to be small enough to actually be accepted by the recipient cell. We've got non-specific acceptance by bacterial cell of small fragments of what we call soluble DNA from the environment. That is the definition for transformation. This is facilitated by some DNA binding proteins on the cell that can actually help to capture DNA from the surrounding area or medium. The cells are going to be able to accept this material. We're going to call those cells competent. That means they can accept this material. Gram-positive and gram-negative species are both capable of this. We're actually able to harness and induce bacterial transformation in genetic engineering, and we call that transfection. And we can make a cell competent by doing things like running electricity through it, making the cell pores larger and that type of thing. All right, so transduction. This is where we're using a bacteriophage to act as a carrier. It's going to pick something up from one bacteria and actually transfer it to another. We're looking here at generalized transduction. There's also specialized transduction. We're not going to focus too much on the differences between the two. So the bacteriophage, as I said, serves as a carrier from the donor cell to the recipient cell. It's going to carry DNA. That's our transduction. We've got the two types, I said, generalized and specialized. In generalized, we're going to have random fragments of disintegrating host DNA that are going to be picked up here by the bacteriophage. Okay, so the bacteriophage is injecting its DNA, and this is the donor or host chromosome here, and it has injected its parts, and that's going to be incorporated. Its DNA is going to be incorporated with part of the DNA of the host cell here, okay, during replication. And so that's going to then, when you get to assembly, you're going to make these mature little bacteriophage, and they now have the original DNA from the bacteriophage and portions of the host DNA in the bacteriophage. So when they go to another bacterial cell and inject it, they are also transmitting part of the DNA that they picked up from the host cell. So pretty much any gene from a bacterium can be transmitted this way. This is something else that we can harness and use in genetic engineering. So briefly going over specialized transduction, you've got a highly specific part of the host genome that's going to be incorporated into the virus. This is actually explained by the existence of that temperate prophage that's going to be inserted into a certain site on the bacterial chromosome. It's going to then become activated. The prophage DNA can actually separate from the bacterial chromosome, carrying a little bit of the host genes with it. This is where we're going to want to look back at our lytic versus our lysogenic cycles for bacteriophage. During the lytic cycle, these viral host gene combinations are going to be incorporated into the particles and then carried to another bacterial cell. Again, you don't have to know all the details of this one, just very briefly. So mutations are changes in the genetic code. They can be both positive and negative. While a vast array of them are going to be deleterious or bad, negative changes, many of them are actually positive. It's the change that is going to be the driving force for evolution, for natural selection to act on. Any change to the nucleotide sequence in the gene is going to be a mutation. Sometimes that mutation is going to cause a change in the phenotype if it changes the amino acid structure enough that it changes the overall structure of the protein. And sometimes it's going to be what we call a silent mutation, where it does not affect the structure of the protein at all. So this can involve the loss of base pairs, adding base pairs. It can rearrange the order of base pairs. We can substitute, delete lots of different things. So when we talk about mutated organisms or mutated traits, we want to understand the difference between wild type versus a mutant strain. Of course, genetic change, I already said, was the driving force of evolution. It gives natural selection something to work on. Any change in that nucleotide sequence of a genome, of the genotype, is a mutation. This is most noticeable when a genotype change leads to a change in the phenotype, which is what you can see here at the right. You have a white-eyed fly here and a red-eyed wild type fly here. Okay, and so the white eyes are the mutants and the red is what we would call the wild type, which basically you could kind of say was the natural version, the unmutated version. So we've got variants in all of these traits here that can occur. Morphology, remember, morph doesn't mean change, it means physical structure. It can be a change in nutritional characteristics, being able to use or not use a certain sugar. A change in the way that you control your genetic control mechanisms, how we actually turn things on and off. It can be resistance to certain chemicals like antibiotic resistance, temperature preferences, or any type of enzyme functions. So it causes mutations, and there's two basic types of mutations. We can have a spontaneous mutation or an induced mutation. A spontaneous mutation is just a random change in DNA that happens often due to errors in replication. An induced mutation occurs because something else has caused it to happen. This can be due to exposure to common mutagens that are going to be chemicals, UV light, X-rays, physical or chemical agents, anything that can disrupt or change DNA. These are a few of the different categories of mutations. We have a point mutation, missense, or nonsense. So a point mutation is one particular point in the triplet codon. It can be an addition, a deletion, or a substitution of the bases. You can also have what we call missense, and that's going to change the code that leads to a placement of a completely different amino acid that can create a faulty non-functional protein. It can produce a protein that functions differently, or it can cause no change at all. Then we have the nonsense mutation, which changes a normal mutation into a stop codon. So it will give you a very abruptly shut off non-functional protein. And so looking at the picture here, you can see some of the different ones here. Okay, so this is our original DNA. We've got TTC at the DNA level. After that has been goes through transcription, it would be AAG, and then for protein synthesis, we would convert that to lysine. Okay, so a silent mutation, remember we talked about that wobble. Okay, if I change that last letter from a C to a T, and it happens to be coding for the same amino acid, AAG and AAA, both code for lysine. So this is where that redundancy helps us to not have problems because of mutation. So that would be a silent point mutation. That's a silent mutation. It is not going to alter the amino acid structure and therefore the protein structure in any way, shape, or form. Then you have here an example of the nonsense mutation, okay? And so we have, instead of TTC, when it's copied, we wind up with ATC. That would be copied over into UAG, which means stop. And so you have a completely non-functional protein. Here are two examples of missense point mutations. And so this is where you're actually changing the amino acid. And depending on which particular amino acid is changed, that determines if it is going to actually cause a problem or not. Some things you can change one amino acid and wind up with something like sickle cell anemia. That is where there is one amino acid that is changed, and it changes the structure of the hemoglobin protein so much that instead of being circular, it is sickle shaped. It can also change an amino acid that has very little change in the structure. It really just depends on the amino acid that is changed. So here's another look at some different categories. Remember, we have the silent mutations, where it alters a base, but again, it does not change the amino acid, so therefore has no effect. You can have what we call a back mutation, and that's when a gene that has undergone a mutation actually reverses back to its original base composition. And we can take advantage of this in genetic engineering and some other things, even to look at certain mutagens in the environment. We use this with bacteria that undergo these back mutations to identify if we have a mutagen, because we can use a mutagen or cancer-causing agent to induce a back mutation. So it's a pretty handy trick. Then we have a frame shift, and this is where one or more bases are inserted or deleted. And so even though you're only inserting here one amino acid, what does that do to your three-letter codons? Okay, so here's our original one. Here's our three codons. We've got methionine, lysine, phenylalanine, glycine, and then a stop codon. Okay, and that's a very much a simplification. They're usually much longer than this. All right, so what happens? You're going to have methionine and lysine. Well, if you insert this one base right here, that throws all of the rest of the bases off. So all of the rest of the codons are off, and so all the rest of the amino acids are off. And so this will often result in a non-functional protein. So again, this is a good table. It shows you some of the different mutations here, all of which we've talked about. Go through these and read these and study these and make sure you understand these. So DNA has proofreading mechanisms to repair mistakes in replication. The cell has additional systems for finding and repairing DNA that has been damaged. You can have UV damage repair using photolyase. All right, this is photoactivation or light repair through the enzyme photolyase. This is successful for only a small number of UV or ultraviolet light mutation. Cells cannot repair severe, widespread damage, and they will die with that type of damage. We also have what we call excision repair, and this is where enzymes will break the bonds between the bases and the sugar phosphate strand at the site of the error. All right. A different enzyme will then come in, remove the defective bases one at a time, and then the remaining gap is going to be filled in by DNA polymerase and ligase. All right. Mutations are a permanent heritable change that are passed on to the offspring of organisms. Most are spontaneous. Most are not beneficial, but sometimes they are if they create an advantage that natural selection can work on. A small number create variant strains that are going to be more readily adaptable, survivable, and increase reproduction. Okay, so as long as the environment is stable, mutants only comprise a very small percentage of the population. If the environment changes, then some of those mutants will be equipped to survive in the new environment at a higher rate than the others. Things like acquired drug resistance are a clear model for this type of selection and adaptation. So these are some of the cooler applications of what we can do with genetic engineering and bacteria. Two techniques have recently been developed that have made huge strides in manipulation of DNA. These are going to be restriction endonucleases or restriction enzymes and the polymerase chain reaction or PCR. All right, so restriction endonucleases, restriction enzymes actually come from bacterial cells. These recognize foreign DNA and break bonds between nucleotides on both strands of DNA. All right. This is beneficial for bacteria and it is protective in nature. We're actually able to harness this capability and utilize it for recombinant DNA technology. This is like molecular scissors. This is how we can actually cut and paste DNA to genetically engineer an organism. All right. We've discovered that there are hundreds of endonucleases or restriction enzymes that are capable of recognizing and clipping base sequences, and we call those palindromes, okay? Palindromes, which are sequences of DNA that are identical when read forwards and backwards, all right? We're reading it from the 5' to 3', and you can see it over here in this example, GAATTC, and here GAATTC in the anti-parallel strand, okay? So it's going to cut them the same. Or a palindrome, remember, is something like kayak, K-A-Y-A-K. It's the same forwards and backwards, 91019, all right? That's a palindrome. So we can actually order these restriction enzymes online. It's really pretty cool. And they come from, the names of them come from like the particular organism and what they do. So E. coli has some that we get, and that would be like EcoR1 or Hindi 3. And those come very specific organisms, and they cut very specific segments of DNA. So if we know which segments we want to cut, we can order that particular restriction enzyme, and it will cut exactly that particular palindrome that we're wanting to cut. And it can make these staggered symmetrical cuts that we call sticky ends, and that's actually kind of helps us to paste them back together. It's going to cut 4 or 5 bases on the 3' strand and on the 5' strand and kind of leave them like overhanging a bit, okay? And so it's like you can paste them back together where they match up. And it sounds crazy, but it's really basically what it is. And so they kind of have these adhesive tails, and they're going to base pair with complementary tails on other DNA fragments or plasmids. And so a restriction fragment is that little piece of DNA produced by the restriction enzymes, or the restriction endonucleases. When you go and cut that, that's what's left, okay? Then we have the RFLPs, which is restriction fragment length polymorphism, and this is the differences in the cutting patterns of different endonucleases. And so we just have to match those up with the DNA that we want to insert. So here at the top, we can see what happens when DNA is denatured. We break those bonds. It's denatured. The hydrogen bonds are broken and separated into 2 strands. 2 regions can then be artificially rejoined. Okay, you can call it artificially rejoin them. This doesn't need to be necessarily from the same organism as long as the nucleotide base pairs match. All right, so we're able to utilize this. The middle here shows how certain endonucleases cut specific bases. And so here's this EcoRI1. All right, so it's going to cut that segment. All right, Hindi 3 is going to cut this particular segment. HAE, that's going to be like from Haemophilus influenza. So this is particularly bacteria that have these in their little toolbox, and we're just borrowing their little molecular scissors to cut these off the way we want, okay? And so the bottom here shows the whole process. You've got a restriction endonuclease is going to recognize here, and it's going to cut that palindrome, CTAG, GATC. That's a palindrome. It's going to cut those sticky ends, all right? And so we're going to then be able to put in something from another organism, which is what it is showing over here, okay? So you can paste in, you can cut the same code from another organism, the same locus on a gene from another organism that maybe perhaps tells it to produce insulin. All right, so this could perhaps be a way, and I'm not saying this is the way that we have made bacteria produce insulin, but this is a possible way that they could have done it. You can take, maybe bacteria had at this area a gene to produce lactase, and we need them to produce insulin. It's the same length, and so it's the same portion of the gene, the same locus, and so we're going to cut that off, okay? And then we're going to be able to paste in the gene to create insulin, okay? So it sounds very simplified, and it's a little more technical than that, but it really is basically molecular cutting and pasting. It's really cool. All right, so here are some other important terms, and like I said, vocabulary is very important. So ligase, remember that's our glue. This is necessary to seal or glue together the sticky ends. It's used in splicing the genes of those plasmids and chromosomes back together. Then we have reverse transcriptase, and that has a role in the replication of the AIDS virus, the retrovirus, and is used to convert RNA into DNA to make what we call cDNA, which is complementary DNA. Remember, I said you could actually read it backwards in some cases. And so cDNA is going to be made from RNA, tRNA, or rRNA, and this is actually used to make eukaryotic genes from an NRA transcript. From a free intron. Remember, an intron is a sequence of bases from a gene that doesn't code from a protein that's been pulled out. So the polymerase chain reaction is a very handy lab technique that is used to amplify DNA sequences. This method is going to use short DNA sequences called primers to select a portion of the genome to be amplified. And so basically you are going to be able to quickly replicate DNA from a few copies to a billion copies, and this has many, many uses. We use it to do DNA fingerprinting. We can use this to diagnose an infection from one single copy of a gene. We can do this to purify certain things. And what you see over here in this picture is here's our copy, okay? So we put it through a polymerase chain reaction. We split it apart, copy it, we make one. Okay, and then basically you're going to do this and you're going to replicate until you have, from one cycle, you've got 2 copies, to over here. I'm sorry, I'm trying to look at it and use my mouse at the same time. To this cycle, you've got 2, 4, 6, 8, 10, 12, 14, 16. And that's a great simplification. All right, time for another practice question or a review, however you want to look at it. So a client is being treated with recombinant insulin, Lispro, and the nurse is asked to explain what is meant by the term recombinant during their teaching session. Yeah, these are really good for you to understand why you need to learn this stuff, okay? So, because you got to be able to explain it to patients in a way that they understand what's going on. Which statement should the nurse make to the client? A, the type of insulin is similar in structure to the insulin that is produced in your body. Sorry for the typos. So there is less likelihood of developing a reaction than if a different type of insulin was used. B, DNA recombinant medication utilizes genes that were transferred from one organism to another to cause a desired effect. C, DNA recombinant medications like Lispro is the same as other insulin preparations but has a longer onset of action. Or D, Lispro will reduce the need for frequent AccuCheck testing during a 24-hour period. So stop and come up with your answer. So the correct answers here are A and B. Your goal here is to explain what recombinant insulin is. All right. So it is A and B. This is showing you an application of the polymerase chain reaction, what you could do next, all right? You could take your PCR, your amplified DNA, and put it into gel electrophoresis. And this produces a readable pattern of DNA fragments, okay? So samples are placed into these little wells or compartments here in a soft liquid agar gel, and then you're going to apply an electric current through them. The phosphate groups have negative charges, which cause the DNA DNA to move towards the positive pole in the gel. A larger fragment is going to move more slowly. Smaller fragments are going to migrate more quickly. And so the position of the fragments is going to be determined by staining the gel. And that's going to give us a genetic fingerprint. So we have sequence maps or genetic fingerprints that give an exact order of bases in DNA or plasmid, chromosomes, or an entire genome. So this is really very helpful. This could be used to determine paternity, for instance, okay, or what type of disease you're looking at, if it's got a particular chromosomes in a pattern you're looking for. Lots of applications here. So the goal of recombinant DNA technology is to remove genetic material from one organism and put it into another so that we can produce things like hormones, enzymes, or even vaccines. Very, very handy. Very useful in things like gene therapy, where we replace a faulty gene responsible for a disease with a gene from a healthy organism. So this is something that is growing right now, okay? It's based on the idea that the error in protein production from a defective gene is overcome by the insertion of the correct gene from a healthy cell into the patient's tissues. We also have CRISPR, which is a little bit more technical application that allows scientists to cut organisms' DNA exactly where they want to.